Hey everyone, we're going to continue with biosynthesis of those small metabolites that are nitrogen containing, like our amino acids, nucleotides, and other related molecules. Last lecture, we looked at amino acids and some of the biosynthetic products of amino acids, like neurotransmitters. But today, we're going to focus on nucleotides, another source of nitrogen that can be utilized and is highly utilized in organisms. Now, a reminder of the naming of nucleotides, they can be separated into purines and pyrimidines. And in more complex multicellular animals, they're often both produced in the liver. They both contain a sugar and a phosphate, but have nitrogenous bases that are different sizes. Remember, these are the A, T, C, and G bases. So because of these different sizes, the two groups are synthesized in two different ways. Now, all nucleotide synthesis uses the phosphoribosylpyrophosphate, or PRPP, which is what donates the ribose and the phosphate part that's necessary for the nucleotide. This is the PRPP. You can see it here. Ribose and phosphate that are shown together here. Now, nucleotides can be synthesized de novo. That means from the beginning, from scratch, and they can be made from amino acids, ribose 5-phosphate, CO2, and ammonia. So just like literally taking the base molecules that we can cobble together a nucleotide. Nucleotides can also be salvaged, this is called the salvage pathway, from RNA and DNA, and also cofactors like ADP, CTP, any of these cofactors. And these can be degraded back down into our base nucleotides. Now, many parasites like malaria, they actually lack the de novo biosynthesis abilities, and they rely exclusively on salvage, making them one of the reasons they are a parasite. So they require the need for already made RNA, DNA, or cofactors that they can break down and salvage the pieces. So compounds that actually inhibit salvage pathways are usually promising antiparasital drugs. So during the de novo biosynthesis of nucleotides, the bases are synthesized while still attached to ribose, usually from two precursors, glycine, which is the precursor to all of the purine nucleotides, and aspartate, which is the precursor to pyrimidine. So again, a very big role for glycine and aspartate. We saw from amino acids that glutamate, glycine, and aspartate are some of the most highly used amino acids. Nucleotide pools of just the regular nucleotides are usually kept low so that cells must continually synthesize them. This is to stop any feedback inhibition, right? If there are big pools of nucleotides, we wouldn't continue to synthesize them. This synthesis, the rate of synthesis of nucleotides, can actually limit transcription and replication. So if you're trying to replicate a piece of DNA, you need to make nucleotides. If you're not synthesizing them fast enough, it can inhibit these processes. So let's look at one nucleotide here. This is the nitrogenous base ring, and you can see where each of the actual elements in the ring are coming from, what molecules they're derived from. This information came from a really cool isotopic experiment where molecules were labeled with different isotopes And finally, when visualizing the final nucleotide after de novo biosynthesis, they could see where the pieces ended up that came from certain original molecules. So you can see, for example, formate gives some carbons. Aspartate is the donator of the nitrogen. That's key. So the amino acid aspartate donates the nitrogen here, and then glycine donates our other nitrogen and some carbons here. Glutamine, an amide from glutamine, is also donating nitrogens into the ring. So all of our nitrogens are coming from those amino acids, glutamine, glycine, aspartate. And then we have some carbons donated from formate and CO2. Adenine and guanine, that's A and G, are our purines. And they are synthesized first as AMP, that's adenine monophosphate, the whole nucleotide with one phosphate on it, and GMP, or guanine monophosphate. They're the production of IMP from those precursor phosphates. I'm going to remind you that PRPP is what our precursor is. That's the sugar with phosphates on it with no nitrogenous base, right? And you can remember that there are 3 phosphates on it for the 3 Ps. 1, 2, 3 phosphates, and 1 ribose. So the letters actually show its structure: phosphate, ribose, phosphate, phosphate. So again, our purines first are derived from that PRPP into an IMP. And these require, the synthesis of the base then requires glutamine, glycine, aspartate, and ATP, among other things, as I just showed in that isotopic labeling. The synthesis begins with a reaction of our PRP with glutamate. It continues with the addition of glycine, and then finally with a full purine ring in the IMP anicetate form. So the anicetate monophosphate is, again, this precursor purine, and it is the base purine ring before we have done our last derivation steps to turn it either into our adenine or guanine. You can see again the steps of this as we're starting with that PRPP, slowly adding on. We start with our glutamine, we add in the glycine, and then we add in folate as well, continued by additional glutamine, another glutamine addition, and then finally the aspartate addition, as you can see as it goes through. Finalizing here in our anicetate monophosphate. So all of this is occurring on that backbone of the sugar with the phosphates. I'm going to go all the way back to show you. Here's our original PRPP. Remember, it has 3 phosphates. 2 of those phosphates are where our base is going to be built, and they act to help empowering this structural building of the nitrogenous base that is being actually made. In the end, we end up with this anicetate monophosphate, which then can be converted into our adenosine or guanine. So AMP or GMP can be made from the anicetate. You can see using, in the case of the adenine, we use more aspartate and fumarate. And in the case of the GMP, we actually use more glycine and some ATP. Purine biosynthesis specifically is regulated by four major mechanisms: the glutamine PRPP aminotransferase, which you can see down here. Glutamine PRPP aminotransferase is one of the first steps in taking our PRPP precursor. And before we get to the IMP, this is one of the major enzymes in starting off that process of de novo synthesis, and it can be inhibited by the downstream products. So if there's a lot of IMP, GMP, or AMP, any of these, it will inhibit that enzyme. GMP and AMP can also inhibit other phosphorylation steps further through the process. Again, this is a lot of feedback inhibition where the products are actually used to inhibit enzymes further back in the process. Now, pyrimidines are the nucleotides that include cytidine, uridine, and thymidine. The synthesis of all of the pyrimidine nucleotides actually begins with the synthesis of the U or uridine pyrimidine, and then this can be derived into the C or T, or cytidine and thymidine. This reaction also uses glutamate and aspartate. It uses bicarbonate and two ATP molecules to provide energy. Again, it also uses our PRPP, which provides that base structure. So unlike purine synthesis, this is very key, pyrimidine synthesis actually starts first by making the ring. Remember, with purine synthesis, we actually had to start with that PRPP portion and then build the ring on it. For our pyrimidines, we actually build the ring first, and then we're going to put it onto the base. So you can see it actually starts with aspartate. Aspartate is the base, and then eventually is cyclized through a process of many enzymes, including the use of our ATP. You can see that after the ring is formed, it is actually then placed onto our PRPP right in this step here during the ornate phosphoribosol transferase enzyme is where we place it on, and then finalize by making our UMP. UMP is then phosphorylated to UTP. Remember, this is just adding on more phosphate, so it is now uridine triphosphate, has 3 phosphates on it. And then it can be converted to CTP if needed. So pyrimidines are regulated also by some of their products, in particular the ATCase. I'll show you this. Enzyme is regulated. Sorry. This is in the formation of our from U to C by the end product CTP, and it is actually accelerated by ATP. Now, we didn't talk about our T thymidine nucleotide. The thymidine nucleotide, which is important for DNA synthesis, is actually made in a roundabout way from our UTP. And it's actually made by deoxy UTP. Now I'm clarifying here all of the other nucleotides we just talked about because they can be used in ribose or made on, or excuse me, because it can be used on RNA or made on ribose. remember that nucleotides used for DNA have to be made on deoxyribose. UTP, regular UTP with ribose, is used in RNA. But if we needed to make the thymidine nucleotide, it needs to be made on deoxyribose because it is only used in DNA. Remember, U is only used in RNA and T is only used in DNA. We actually have to make UTP first on deoxyribose and then make it into our TMP again on deoxyribose. So it is in this very roundabout way. So first the deoxyuridine triphosphate is made, and then the deoxyuridine triphosphate is made into deoxyuridine monophosphate, which looks like the word dump, DUMP, by a molecule called deoxyuridine triphosphatase. It just removes the phosphates, goes from a triphosphate to a monophosphate. And then the kicker is to go between deoxyuridine monophosphate to deoxythymidine monophosphate, you need this specific enzyme called thymidylate synthase. This actually adds that methyl group on to make the T nucleotide. Thymidylate synthase is a target for some anti-cancer drugs, and this is a very specific reason. Why would we want to stop methylation, adding a methyl group on, just like in this case, going from U to T, we add a methyl group. So why would you want to stop methylation in general in cancer drugs? It's a question I'm posing to you to think about. So again, I wanted to show that outline. Remember, we can make UDP and then we can also make CDP. These are both used in forms for RNA. They can be converted to their deoxyribose forms, DCDP, DUDP. They can then be phosphorylated to be the triphosphate form. So this is deoxycytidine triphosphate or deoxyuridine triphosphate. Either one of these, they can be converted into each other. So C can go back into U. But ultimately, we have to get deoxyuridine triphosphate to then form our deoxyuridine or thymidine monophosphate. Here is an outline of the actual enzyme, again, that important enzyme that is doing the transferring, the methylation of our deoxyuridine monophosphate to deoxythymidine monophosphate. It is connected to another enzyme, which is serine hydroxymethyltransferase. This is taking, you can see, serine from glycine and using tetrahydrofolate. Folate is often categorized as a vitamin molecule. And this process is where we get the methyl from. The methyl can then be transferred onto the, from the uridine and then on to make the thymidine. So it's through this folate molecule, tetrahydrofolate, that we actually can do this transferring of the methyl group. So when you have folic acid deficiency, this can reduce thymidylate synthesis. When, especially in areas with nutritional, poor nutritional habits, or they are not able to get folate from nutritional sources, there can be reduced thymidylate synthesis, which causes uracil to actually be incorporated into DNA instead of the thymidine. So U instead of T because we can't make tea if you don't have folic acid. There are repair mechanisms to remove uracil by creating strand breaks. And this can, of course, affect structure and function of DNA, which is associated with cancer, heart disease, and neurological impairment. So ultimately, if you have folic acid deficiency, you can end up accidentally incorporating uracil into DNA. And then when trying to remove that uracil, potentially causing more damage that ultimately can lead to mutations and disease. So let's talk a little bit about the ribonucleotides versus deoxyribonucleotides. We've kind of touched on it a bit. Remember that nucleotides, generally, except for thymidine, are initially made with ribose as the sugar, which is in RNA. But DNA, of course, uses deoxyribose, right? So we looked at our thymidine monophosphate is the only one that has to be made in this special way because it is made on the deoxyribose. All the other ones are formed on ribose, but then can be made into their counterparts on deoxyribose. Remember, deoxyribose is essentially the same as ribose, except it is missing a OH, a hydroxyl group, on the sugar component. So to go between our deoxyribose and ribose connection, if you wanted to make a ribose into a deoxyribose or vice versa, you actually ultimately need a lot of NADPH and glutathione. This is through, as you can see, either two mechanisms, one starting with NADPH, glutathione, and then glutaredoxin, which can help regenerate the enzyme that allows for the processing of the sugar from ribose to deoxyribose. Or you can use, instead of our glutathione, you can use FAD and thioredoxin as well. This is the flavoenzyme with FAD as a cofactor. The actual enzyme ribonucleotide reductase, it's shown here, is a large subunit, as you can see, that utilizes all of these components. It has to be able to not only do the regulation of the sugar, but utilize many different cofactors, including potentially glutaredoxin, glutathione, and NADPH to go through this process. Now, this has a double regulatory subunit in which it is generating radicals. It contains iron to do this and uses a tyrosine to abstract a hydrogen from sugar. So it's actually doing this through radical chemistry within it and using tyrosine as the part of the enzyme, the amino acid on the enzyme, to stabilize that radical electron. This is just a quick overview of what's happening during the mechanism of ribonucleotide reductase. Notably, I just wanted to show that it does use radical chemistry stabilized by a tyrosine within the enzyme. Now, because ribonucleotide reductase is so important for the transference between the deoxy and ribose form of the nucleotides, it is the area that is most regulated. And you can see there's a lot of regulation going on between the forms of the sugars themselves, the deoxy form or the the normal form, that can then lead to regulatory, either turning on or off of the enzyme to transfer between the two. So let's talk now a little bit about the transformation of nucleotides into other molecules and their use. Purines specifically can be catabolized into things like molecules like hypoxanthine, xanthine, and ultimately uric acid. This occurs through dephosphorylation and deamination. We have to remove that nitrogen out. And again, ultimately into uric acid, which can be excreted. Now, this is mainly occurring through a enzyme called xanthine oxidase, which actually spiders and other arachnids lack. This is why their uric acid form can't be produced in these animals. So some fish, for example, will actually excrete more nitrogen as ammonia than as urea. Uric acid can be converted into other molecules as well, other metabolites, such as allotonin, which can be formed in most mammals, allotinate, which is only formed in bony fish, and then further broken down, potentially, as I said, into urea, which is only seen in marine invertebrates. So the degree of further oxidation of uric acid is very organism dependent, how much further down you can break it down. Now, us as primates, birds, and reptiles, and many insects, we excrete uric acid, but again, many other organisms can further break this into ultimately, for example, in fish, down all the way into ammonia to excrete. Uric acid can build up in some cases. A particular disease that's known for this is gout. This is shown by the symptoms of painful joints, often in toes, and this is due to deposits of sodium urate crystals. It primarily affects men, and this is thought to be because of the involvement of a genetic under excretion of urate, and may involve overconsumption of fructose, the sugar. One of the treatments is to avoid purine-rich foods because, as I just noted, purine is broken down into uric acid. So to kind of limit the production of uric acid, purine-rich foods such as seafood and liver, if you were to eat something like pate, have to be avoided. It can also be treated with the xanthine oxidase inhibitor, allopurinol, which again is going to stop this conversion of nucleotides or purine nucleotides, AMP and GMP, into uric acid, right? They would stop at these other forms, these other metabolite forms, and have to be converted into something else. Now, hypoxanthine is a normal substrate of xanthine oxidase, but allopurinol, that drug I just noted for patients of gout, as you can see, is a drug that mimics almost exactly hypoxanthine. So it binds as an effective inhibitor into the xanthine oxidase enzyme and is instead converted into oxypurinol, which is a competitive inhibitor that remains tightly bound to the reduced enzyme. So Allopurinol is not the competitive inhibitor, but it will bind xanthine oxidase, and then oxypurinol, when it is converted, will stay in the xanthine oxidase after it's converted inside. Breakdown of pyrimidines leads to ammonia and urea, and it also can produce intermediates for the citric acid cycle. So for example, thymidine can be degraded into succinyl-CoA, a main citric acid cycle intermediate. Now, as noted at the very beginning, purine and pyrimidine bases can also be recycled by the salvage pathway. And instead of being broken fully down into uric acid or ammonia, they can be utilized again and salvaged for other nucleotides or things like ADP, AMP. The brain in particular is very dependent on things like salvage pathway, because the lack of the hypoxanthine and guanine phosphoribosyl transferase in particular can lead to a disease called Less9 syndrome. And this is a neurological impairment, and it actually results in finger and nail biting. So this enzyme, the hypoxanthine guanine phosphoribosyl transferase, again, is part of this salvage pathway. And because the brain is so reliant on it, if there is any type of problems with those enzymes, it can lead to neurological diseases. There are many chemotherapeutic agents that target nucleotide biosynthesis. Glutamine analogs, they can actually inhibit glutamine aminotransferase. There are fluorouracils, which can be converted by the salvage pathway into a fluorodeoxyuracil monophosphate, which actually will inhibit the thymidine synthesis. There's methoxytrexonate and amino paterpin, which will inhibit the use of dihydrofolate reductase. Again, this is going to stop formation of the deoxythymidine monophosphate. You can also look at antibiotics that target nucleotide biosynthesis. We just looked at allopurinol because of its similar composition, and also trimethoprim, which can actually inhibit bacterial dihydrofolate reductase, but it binds human enzyme of the same enzyme of the dihydrofolate reductase, just a little less strongly. So these are all types of these drugs that use nucleotide biosynthesis and those enzymes in nucleotide biosynthesis as targets. All right, well, that is it for nucleotide biosynthesis and our general biosynthesis of those small nitrogen-containing molecules. Next up, we're going to look at hormone regulation of small molecules in the body.